Tate Chronicles now transmitting.
Welcome to the Tate Chronicles on Healthcare Now Radio. And now, here's your host, Jim Tate.
Good day, citizens of the free world, from border to border, coast to coast, and to all the ships at sea. I bring you a warm welcome. This is your correspondent, Jim Tate, and thank you for tuning in today to the Tate Chronicle. Join me as we cut through the fog that exists at the leading edge of healthcare technology and culture. I'm really excited. My guest today is Greg Archambeau. Is that close enough? Okay.
There you go,
Archambeau,
Jim.
Who's the president of
Revalesio,
which is a clinical stage pharmaceutical company based in Tacoma, Washington. It will be a unique conversation because they have received a FDA fast track designation for the treatment of both ALS and acute ischemic stroke for their novel foundational compound process, RNS-60. And they're now preparing to launch a phase 3 clinical trial in stroke based on their phase 2 results. So, Greg, thanks for joining me on this episode of the Tate Chronicles.
Jim, thank you for the invite. Looking forward to today's discussion.
So, Greg, the first thing I want to ask you to do is educate me a little bit. From what I understand, the technology behind RNS-60 is using fluid dynamics to biologically activate a saline solution. What does that mean? How was this discovery made, and how did it move from concept to a clinical candidate?
Yeah, Jim, again, great question. So RNS-60 chemically is oxygen, specifically dissolved oxygen, supersaturated in saline. So the process in which we do that, though, is a well-understood fluid dynamics process when you're trying to mix gases and liquids. And so we used this technology early on within agriculture research. And what we saw was an increase in yield in agricultural crops, as well as a resistance to disease stress in agriculture crops. And our founder, Eric Russell, at the time went, if this is working in plants, what would this do in humans, in human biology? And that was the beginning of our journey almost 20 years ago with this product.
So let me take a stab in the dark here, Greg. So saline solution, supersaturated with oxygen in some way. And in terms of a ischemic stroke, which means blood flow has been cut off to at least a portion of the brain, is the treatment involved just really flooding that zone with oxygen to prevent further damage?
So that's a part of it, Jim. A part of it is the supply of oxygen. But we see a different effect than if you would say standard oxygen treatment during stroke. You've seen individuals or companies look at normobaric oxygen treatment, so just inhaling oxygen, or even the use of hyperbaric oxygen chambers. There's been a lot of animal research done with a hyperbaric oxygen chamber. We see a different response with RNS-60 than other treatments with oxygen. And we believe this is due to a signaling component within the body that engages pro-survival pathways within neurons. And so it helps the neurons survive that initial shock, that initial stress associated with the clot that forms that ischemic stroke. So we use other oxygen therapies as controls against RNS-60, and we see a different effect with RNS-60. So there's something about the processing, the fluid dynamic processing of when we saturate the saline with oxygen that's giving this different effect.
And so in terms of what's happening at the cellular level, is inflammation reduced? We're not only talking about the ischemia, but is inflammation reduced?
Yeah, there's two components to it. I talked about the pro-survival pathway with neurons. So we're helping keep neurons alive in that stressful situation. But we also see a reduction in inflammation, not what you would see with, say, a steroid. It's more of a tampering of the inflammation. And so we believe that that's important. And we also see, again, promotion of survival and function in other cells involved in the cerebral vascular system that is all really under attack during an ischemic stroke.
So first let's talk about the clinical trial. So I believe you've completed a phase 2 clinical trial, correct?
That is correct.
Well, without giving away any intellectual property, what were the results of that phase 2?
The results were fantastic. So this was our proof of concept study. So we had 82 individuals that received either placebo, a high dose of RNS-60, or a low dose of RNS-60. RNS-60 is delivered for 48 hours via an IV.
Okay.
And what was unique about the design of our study is we used imaging, MRI, as a biomarker for our success in stroke. Because typically what you're doing is you're looking at are individuals independent or dependent 90 days after their stroke. You typically need to enroll 500 to 1,000 patients to see an effect on that functional endpoint of how well is the patient doing. We designed this proof of concept study to look at the size of the stroke immediately after best standard of care. So everybody that was enrolled in this study received what's called mechanical thrombectomy, where they pull the clot.
Yes.
And we took an MRI right at that point to establish a baseline for the size of the stroke. And then we took an MRI 48 hours later. And what we saw in comparing high-dose RNS-60 to placebo is 50% less brain volume loss during those 2 days post-stroke. That was the huge win in our study, is that we're showing a physiological difference in the brain, less brain loss with treatment with high-dose RNS-60 in 48 hours. And then we looked at, did patients have a good outcome at 90 days? And we saw that 63% of individuals had a good outcome versus on placebo, it was 46. So quite a big difference as it relates to individuals that were independent at 90 days.
So doing a clinical trial, normally there's a big recruitment effort. But I guess you were doing clinical trials with people coming into emergency departments having real strokes.
Absolutely. It's a very dynamic situation to be in.
Yeah. And
I think we did a great job, our operational team, of designing a protocol that fit into that dynamic treatment environment where individuals are coming in, they're getting assessed for mechanical thrombectomy, where they enter through the groin and remove the clot. And time is brain. So that surgery has to happen as quickly as it can.
Yes.
And we provide RNS-60. We actually manufacture it here in Tacoma, Washington. We provide it in ready-to-use sterile IV bags. So it's very easy for the clinical team to incorporate that either while the procedure is going on to remove the clot or immediately at the end of the procedure to get treatment going.
You say you have these, I guess, IV bags that are kind of ready to go. What's the storage requirements? Have to be kept cold? Yeah.
It's a cold chain product. I'm glad you brought that up. It's a cold chain product with the stability out to 2 years.
And so this is infused over at least during the trials, it sounds like, a 48-hour period, correct?
That is correct. We're going to keep that same infusion rate in the phase 3 study, a 48-hour infusion.
You kind of brought it up. When does the phase 3 trial begin? How's that going to go?
Well, Jim, great question. We're currently raising capital to execute that phase 3 trial.
I know they're expensive. They're very expensive.
They are expensive. That is correct. What's been great, though, is we've already been able to select 30 sites 30 clinical sites in the United States to participate in this phase 3 trial. Sites are pretty excited about being a part of it for a couple of reasons. One, we have the data showing reduction in brain volume loss. So unlike some other individual or companies that are in the space, they don't have that biomarker of success in their small proof of concept study. They're just looking at functional endpoints at 90 days. And it's hard to imagine, how are you going to show a benefit? How more individuals that are doing well at 90 days, if you aren't decreasing the size of the stroke after best standard of care. A lot of people don't know that, but the stroke is still happening even after you've reestablished blood flow. And there's even injuries being occurring as that blood flow is reestablishing, because you're now distributing damaged cells, a lot of damaged material to that area of the brain that needs the blood supply provided. So you have to do that, but there is damage that's being done during that process.
Absolutely, it's a cascade of events. It's a process.
100%. There's a great publication last year from Jeff Saver, one of the leading voices in the field out of UCLA, where he describes there's really 6 injuries that are happening during a stroke. And those 6 injuries are a cascade that occur over about 24 hours. And so that's another thing I think that's excited the field about RNS-60 is we have data that shows in 5 of those 6 areas of injury, RNS-60 has an effect. And we also have an effect across that full range of 24 hours post-stroke. Not a lot of companies have that type of data. They're focusing more on maybe the first 3 or 4 hours post-stroke because their drug only works on neurons. But our drug supports mitochondrial structure and function in all the cell types that are in the cerebral vascular system, which gives us a larger window to have an effect on a stroke patient, which again is one of the reasons why the sites are excited about our product, because it's not as an acute treatment as some of the other products in the field. There's more time available for it to work.
How many, I guess, like a better term, participants do you expect to have, or do you want to have in this phase 3?
So we've powered the study. So we've designed the study for 620 patients, and that will give us statistical significance on that functional endpoint that FDA wants to see to review a new drug application. Other companies in the space have had a smaller effect size on good outcomes and have run 1,000 to 1,100 patient studies. So the 620 is one that we're confident we can execute on in a timely fashion.
In the news over the last couple of months, I've heard more and more about the phrase, the FDA fast track designation. How does that affect y'all? And I believe you've been approved for it. So what does that mean? And is that good for everybody?
Yeah, it is. I mean, fast track designation gives you more opportunities to interact with FDA. So that's one of the main benefits, I think, is having additional conversations with the agency.
And during a trial, as things arise, does that mean you can kind of communicate? And I'm not quite sure what word to use, but communicate with the FDA for modification in the trials if unexpected things start to occur.
That option.
That
option exists. I don't think, obviously we're going to be blinded as well as the clinical sites will be blinded. Even the researchers that are reviewing the MRIs to determine the size of the MRI or size of the stroke immediately post mechanical thrombectomy and then 48 hours later, obviously all of those individuals are blinded. So I think the need to interact with the agency in the midst of the study, I don't believe we'll have that. In our proof of concept phase 2 study, we had great safety data. So we don't expect anything to come up as it relates to safety, but obviously we'll have a data safety monitoring board as part of that phase 3 study. So the option exists, Jim. I just don't think that I don't see us needing to use it because we're essentially taking our phase 2 protocol and just executing it in more patients. So we've got experience in this. The clinical sites have experience in executing this protocol. So we really think it will be a seamless study.
What's the timeline on it?
Yeah, the timeline is it's going to take about 2.5 years from first patient in to last patient dosed.
And in this particular case, are you asking the FDA for approval based on indications for ischemic strokes and ALS?
So the filing with FDA at the end of this phase 3 study would be for ischemic stroke. We would. This will give some insight, I guess, to the strength of our phase 2 data. If we even saw half the effect size that we saw in our phase 2 study at that 90-day, did a patient have a good outcome or a bad outcome?
Sure.
If we even saw half that effect size, we would still move forward with filing because it would mean a new standard of care in the field of treating ischemic stroke. So our filing would be ischemic stroke specific. We've got great data in ALS, but we would need to execute a phase 3 study in ALS, repeating what we saw in our phase 2 to be in a position to consider filing for ALS.
Sure. Let me mention to our audience, if you're just joining us, I'm Jim Tate, and you're listening to this episode of the Tate Chronicles. I'm speaking with Greg Archambeau, the president of
Revalesio.
Revalesio. Thank you, Greg.
Yes.
And so, other indications. So, other indications, potential that has been thought of outside of really protecting the brain, other parts of the body.
Yeah, well, this goes back to what RNS-60 does. We know RNS-60 helps stabilize mitochondria under stress. Yeah. Actually, you bring up ALS, in probably within 3 or 4 weeks, we're going to have a publication in Muscle and Nerve that shows specifically the effect we have on the structure and function of mitochondria. So we've got the opportunity to look into other diseases or associated with organs or areas of the body that have a high concentration of mitochondria. And so that could be acute kidney disease is one that comes to mind that we could look at. We've got solid preclinical data in Alzheimer's and other chronic neurological disease conditions. But currently our focus is 100% on stroke. Sure. And generating that phase 3 data and being in a position to file a new drug application with FDA.
You know, Greg, the current treatments of acute ischemic strokes, it's kind of a nightmare when you hear somebody's had a stroke because, the brain's involved. Right now, there's a relatively narrow treatment window. Well, of course, you want to come into that treatment window as soon as possible. But even if it's delayed for a period of time, is there still potential benefit? Like if it's 4 hours before somebody says, oh, we probably should start this.
Yeah, that's another great question, Jim. When we designed our phase 2 study, we designed it with individuals that were receiving best standard of care. So they all received a mechanical thrombectomy where they go in and pull the clot. That procedure can be done up to 24 hours after you've had a stroke. We wanted to establish as wide of a safety time window for the use of RNS-60, and we didn't want to slow down enrollment by focusing on, let's say, less than 12 hours since enrollment or since stroke.
Sure.
So our proof of concept data was actually generated from individuals that had a stroke within 24 hours. Our average time since stroke was in the 9-hour time frame, but we had individuals that were further out than that. So we're actually showing that our drug, because again, it works on mitochondria in any cell type within the cerebral vascular system, that we've really widened that window for potential treatment. Now, when we looked at a shorter window in our phase 2 study, so we carved the data a different way and looked at those individuals that had had a stroke within 12 hours. the average time in that group was around 6 hours. Our effect size got even stronger. So that's another thing that's really encouraged us about our data is everywhere that you look, every way we look at the data, it supports what you would expect to see.
Mm.
If we're able to intervene earlier, we should see even a better effect of the drug. And that's what we saw in the functional outcome with those individuals that had a stroke within 12 hours. 72% of those individuals had a good outcome at 90 days, where the average, again, is about 40 to 45% of individuals that have a mechanical thrombectomy have a good outcome in 90 days.
I'm fascinated by the difficulty in doing these clinical trials. So you have maybe a center that is a partner doing a clinical trial. I guess there's a primary investigator there, and if
Correct.
And the ER team has kind of been briefed that somebody comes in showing signs of this, getting in touch with that primary investigator and have some family members sign a document or something. Is that kind of how it all goes?
Yeah, it's amazing. It's been described to me, Jim, as you've got a stroke team sitting on the platform of a train station, and you've got a bullet train that is pulling through that train station but not stopping. You have to get everybody onto that train before it leaves the station. That's how it's been described to me as it relates to the patient, their family, to get informed consent, to get the whole stroke team, and then to get the drug. It's really amazing what those clinical research teams can do at these stroke centers.
One thing I think about, Greg, is patients and their families. When somebody has a stroke, there's often shock and then hope and disappointment and how much reversal all those things are kind of going on. What could you say to stroke victims, stroke patients, or their family members to understand about where RNS 60 stands right now and what a realistic timeline might look like in terms of it being available?
Well, stroke has impacted my life. My dad's had 2 strokes. And so I understand the family side of the equation. And I think I'm proud to be involved in trying to work on a solution for stroke. There's a lot of the field, I would say, is staying away from ischemic stroke because of all the failures in the past.
Right.
But when the best standard of care can only give a good outcome 40 to 45% of the time, there is a significant unmet medical need. And we need, as a community of drug researchers and developers, we should be looking to address that need. And I think RNS 60 is one of the most promising candidates in the field because of the data that we've generated and showing RNS 60's ability to lower the growth of the stroke. Preserving brain has got to be where we start. If we can preserve brain with any of the compounds that we work on, Revalesio or another company, then we've given ourselves a fighting chance of having a good outcome for the patient and the patient's family at 90 days. So from a timeline, I wish we could get into the clinic tomorrow.
Sure.
There is a challenge right now with the investment community very much interested in weight loss drugs, the next GLP-1, or AI-developed drugs.
In silico.
But, but from Revalesio's standpoint, we know where the gaps exist. Stroke is one of them. Alzheimer's is obviously another, where there's significant need and significant impact on our healthcare system from a cost standpoint, where somebody needs to step into that space and take the risk of executing phase 3 trials to potentially get a product to market. And we think we've mitigated that risk about as much as we could at this point in time with the design of our clinical trial, the use of infarct volume growth as an early signal that our drug has the potential to win in a phase 3 study. So we feel very well positioned. It's just a matter of finding like-minded individuals that are willing to take that risk of moving into a space like stroke that is somewhat considered the graveyard of drug development, given failures from 15, 20 years ago.
Greg, we're almost totally out of time. Tell our listeners and tell me how we can get in touch with the company. What's the website? How do we find out more and stay in touch?
Yeah, you can find us on LinkedIn or at our website, www.revalesio, revalesio.com. We have all of our publications on our website. Again, I mentioned that we have a new one coming out shortly in Muscle and Nerve, where there's some fantastic images of the mitochondria in a diseased state and the mitochondria when treated by RNS-60. So that's the best way to stay abreast of what we're doing is LinkedIn or our website.
Right. Well, it's exciting work. And my background keeps me interested in new developments, especially when we're not dealing with toxic compounds, but things like saline and oxygen. So that's fantastic. To our audience, thanks for joining me on this episode of the Tate Chronicles. And Greg, a special salute to you. Thanks for coming aboard today.
Jim, thank you so much for reaching out. I really enjoyed the conversation today.
You can find more information on this show's program page at healthcarenowradio.com. That's healthcarenowradio.com. Until we meet again, here's wishing you smooth sailing and safe harbors.
Tate Chronicles transmission ending now.